In this class, I am going to discuss the prokaryotic gene regulation. Gene regulation means how a gene is transcribed. Basically, the gene regulation means the process of transcription regulation. Okay, nothing else. So from this transcription, the mRNA is formed, and from the mRNA, the protein will be expressed. And this is the total process of gene expression. Okay. So during this type of transcription and translation, some factors are responsible. I have already told you in the class in translation, where you have already noted that there are some factors. Okay, these factors are basically controlling the gene transcription or translation. This is known as the gene regulation. So what is the gene? Gene is the segment of DNA. It is nothing else. It is only the segment of DNA that contains some nucleic acid sequence. Okay. And when this segment of DNA controls the trait of organism, that means the trait means, what is trait? Trait is the phenotypic nature of the organism. And this is totally controlled by the gene. And this can be physical or metabolical. Okay. And the information that is encoded in the DNA strand into and it transcribed into RNA, and then it translated into proteins. And this is the whole scenario is known as the central dogma. That is the flow of genetic information. Okay. And this type of segment, what is transcribed into the mRNA is known as the gene. Okay. In a prokaryotic system, there is only one segment of DNA, and thus this is known as the haploid gene. Okay, but in case of eukaryotic system, the gene may be, the copy of the gene may be more than one. Okay, generally the copy is 2, and in case of higher ploidy level, there is, we can found the more copy number of the gene. Clear? So what is gene regulation? I previously told that gene regulation is a process which a cell determines which genes it will be expressed and when. The time also is a factor. You think, you all know that suppose I am consuming the protein. Okay, I am eating some protein, and that's why I have to digest this protein, and for the digestion of the protein, the protease enzyme is required, and this enzyme is a protein. So for this digestion, the protease must be produced or must be expressed. So when I engulf a protein, the protease will be expressed. So this is the time. When I am not engulfing the protein or not consuming protein, the translation process of protease is stopped or halted. So there is a time when this gene will be expressed. In case of prokaryotic system, the gene is circular. Gene is the form of a circular bacterial chromosome, and the segment of this chromosome is known as the gene. And in this case, a gene has two parts. One is the upstream portion that contains the regulatory portion, and another one is the downstream position where the transcribed portion of gene is situated. That means the downstream position from where the mRNA will be expressed. Okay, from this downstream region, the mRNA will be produced, and from this mRNA, the proteins will be produced. So this is the basic structure of a prokaryotic gene. Okay. The transcription regulation in common mechanism in a prokaryotic, and it can be occurred by two processes. One is positive regulation, another one is negative regulation. So what are they? Positive regulation means the activator is there. What is activator? Activator means a molecule or a factor that activates the gene regulation. And the negative regulation, there is a repressor which blocks the regulatory mechanism. What are they? Suppose in this picture, it is clearly depicted when the operator or the enhancer site, the promoter's upstream region is known as the enhancer. When these two sites are bound with the activator molecule, the transcription happens. Without this activator molecule, no transcription will be happen. Okay. Or in case of negative regulation, this is known as positive regulation. Clear? When the negative regulation comes, the operator is bound with the repressor molecule, and when there is no repressor, then the transcription will be possible. Okay, so this is the negative regulation. And there is another type of regulation known as auto regulation. That means the transcription product or the translation product may be used for the protein synthesis mechanism regulation. Okay, this is known as the auto regulation. That means the same gene product control its regulation. Clear? So this is known as auto regulation. So basically there are three types of regulation. One is positive regulation, another one is negative regulation, and third one is the auto regulation. In prokaryotic system, this negative regulation is very common, and in case of eukaryotic system, the positive regulation is most common. Okay. So this is the RNA polymerase which binds at the promoter region, and the operator site is situated in between the promoter region, and the activator site, activator binding site or the enhancer site is just upstream of the promoter region, and the promoter region is able to bind the RNA polymerase. So binding the RNA polymerase, this gene will be able to transcribe, and you see that this is the operator site which is just located inside the promoter region. Clear? So when this type of when the operator is here and a repressor is bound here, it can compete with the RNA polymerase by the means of competitive inhibition. Okay, so when there is a competitive inhibition, the operator cannot allow this RNA polymerase to sit. Clear? So this RNA polymerase, when doesn't sit here, it cannot initiate the transcription. But when the activator molecule is sits on the enhancer sites, the RNA polymerase binding is enhanced. The binding possibility of RNA polymerase is enhanced, because you think that you are sitting on a tool and you are also sitting on a chair. Okay, so chair have a back side, and you can lean onto the chair, onto the back side of the chair, so that is more comfortable. And suppose there is no activator molecule, so this is supposed to be a tool. Okay, there is no backside, so this backside promotes the comfortable nature of the RNA polymerase. So the RNA polymerase can sit comfortably and it initiates the transcription. Okay, so this is the basic mechanism of gene regulation with the help of positive regulator. This is the positive regulator and this is the negative regulator. Clear? So the transcriptional control of gene expression in prokaryotes. The two studies main mechanism of transcription control of gene expression are the Operon. One is Operon and another one is the cascade of gene expression. In your syllabus, only the Operon is there. So the cascade of gene expression is beyond of your syllabus or scope of your syllabus. So I only describe the Operon system. Basically, this Operon system first developed by two scientists. One is Jacob, another one is Monod in the 1961, and they proposed that the Operon model. The Operon model means in a prokaryotic system, the genes are polycistronic nature. And what is polycistronic? The polycistronic means, just a minute, the polycistronic means there are a cluster of genes. Okay, this is the Operon. This is the Operon. That means with the presence of the regulatory system, there is a cluster of genes, structural gene is present. And these structural genes are not a single stretch of nucleotide. They are interrupted by the stop codons. Okay, so from this structure, from this figure, we can easily identify that there is 4 genes, and A, B, C, D. These are 4 different genes, and they are controlled by the same regulatory mechanism of the gene. So this is known as Operon. Operon is a cluster of genes. Those are regulated by the same regulatory mechanism. Clear? So The operon consists of several structural genes required for the metabolism of lactose or several other type of energy producing elements, and basically they are known as coordinate regulation. Okay, so when we are considering the regulation of these 4 genes together, they are known as the coordinate regulation. And I also told that the repressor binding site is known as operator and site for transcriptional activation is known as promoter. So these are already told. Okay, so the Operon model of regulation. Basically, the operons are 2 types of operons are there. One is inducible operon, another one is repressible operons. What are they? Inducible operons is activated by the small molecules known as inducers. Okay, and these mode of regulation is degradative, like catabolic pathway. When we degrade the molecules to produce the energy, this is known as inducible operon, like the lac operon. Lac operon is an inducible operon. And another one is repressible operon. That operon is shut off by small molecules known as co-repressor. And the mode of regulation is biosynthetic or anabolic pathway, like tryptophan operon. In case of lac operon, we use the gene products of lac operon genes. We use these gene products to catabolite lactose or to degrade lactose. And in case of tryptophan operon, the gene products those are synthesized and used to synthesize that tryptophan. Okay, so there is an anabolic method that is used in case of repressible operon. Another one is inducible operon, where the catabolic pathway goes on. So students, this is the main thing what is by which genes are regulated, and I think this is helpful for your exam purpose. So please ask me if you have any question. Okay.